My talk is going to focus on, I come from a pharmaceutical company, big pharmaceutical company, and what I'll try to do is I'll try to say what is the end point in mind. If you are to go all the way with the diabetes drug, what is it? So it all starts with an unmet medical need. If there's not a need for a drug, we shouldn't develop it. Is there a potential market to actually sell it? How do you develop it? What is the process? What potential targets could you think of if then that should fulfill those unmet medical needs? A key thing is that you keep an eye on risks very early on as you engage in drug development. And then eventually I get into the future, how I think that it may fit well with what is starting here in Qatar. We are an old company from 1876. We are in many ways too old. You are young. We try to get young again by the way we work, and I'll get to that at the back end of this. So first of all, the unmet medical need. I think I heard Her Highness yesterday say 20% prevalence of type 2 diabetes and 20% prevalence of impaired glucose tolerance here in Qatar is really scary. That's staggering numbers and of great concern. Driving that, we know, is genes of great importance, but also the environment. There are a number of things that are underlying diabetes, which is at the bottom of the iceberg, and then eventually it leads to diabetes, which is the tip of this iceberg. But all of these are leading to a number of medical concerns that I'll address later. So I want to introduce a concept which is diabetes progression. So diabetes is not just diabetes, it's actually a progress over time from pre-diabetes leading to eventually failure, where you need maybe not one drug, but a number of drugs, and eventually you may end up needing insulin. And this progression is, as you can see in the bottom curve, is associated with a loss of insulin secretion over time. We believe that if you can prevent the loss of insulin secretion over time, you can stop diabetes. We believe if you could actually boost it to begin with, you may actually be able to prevent diabetes. We know that happens in a number of animal models. It's possible in a number of animal models to cure diabetes. Obviously, there's a leap of faith going into man that actually translates, but we think that that's our final goal, is actually to come up with a treatment that will cure diabetes through that mechanism. So this is a little bit centric from my company, but I think it speaks to many others. At the back end, you have treatments for the complications. So if you've had diabetes for a long time, you get complications. It's important to treat those. Much better would it be if you could avoid the complications because you could avoid the disease in the first place. Late-stage treatments include the insulins. There is a new class of compounds coming in, incretin mimetics. And here you can see we have these products, but we don't have an all anti-diabetic agent. And the reason is it's very hard to find new good targets that address diabetes, and we're desperately looking at new targets. And I know my fellow speakers later will get much more into details on how to define those targets. But Lilly is really in need of finding targets, and especially it would be important if there are genes that could be linked to diabetes here in this region to actually target those with oral agents. So this is our strategic focus. First of all, you cannot register a drug for diabetes unless you can lower glucose. So you have to have glycemic control no matter what you do to the disease and the outcomes. If that's the primary indication, you have to lower HbA1c by 0.4%. We think that treatment should ideally modify the disease. If it just continues to progress, it's just a short fix. I'll spend little time on micro and macrovascular complications, but we have a cardiovascular drug hunting team that is looking at the link between diabetes and cardiovascular disease. And then we have a drug hunting team looking at weight loss, and obviously there's a tight link between obesity and diabetes, and any good treatment for obesity could potentially also be a good treatment for early diabetes if it's linked to obesity. So how do we look at that, and what are the mechanisms that we look at? Well, we have a number of things that are of interest. First of all, you need to get a good anti-hyperglycemic mechanism in place. our highest priority is to actually restore insulin secretion. As I showed you on one of the previous slides, insulin secretion declines over time, and our highest priority is to normalize that if we can. We think that is secure to diabetes if you can do that. The number 2 priority is to reduce insulin resistance in type 2 diabetic subjects. I think there was a time when insulin resistance was the hottest topic, then it was insulin secretion. I think the right balance really is that insulin resistance should get more importance. And then hepatic glucose production, which is the third mechanism of controlling glucose in the circulation, is number 3 priority, in part because metformin is an existing drug and to some extent takes care of that. Ideally, we'd want a drug that does more than one thing, that actually if you have a drug that does all 3 of those, that would be the ideal drug. So this is the diabetes epidemic, and as you can see, these are staggering numbers with a 114% increase worldwide, and in the African Middle East, it's going to be 162% increase from 2000 to 2030, which is just scary numbers. Also, look at the numbers for Africa and Middle East versus the Americas. The total numbers in 2 decades is going to be the same in Africa and Middle East as in the entire Americas. So these are really scary numbers. So it relates to the obesity epidemic as well. And if you look at what happens, what makes this an opportunity for a company like ours is that patients need more and more drugs to get on target with treatment, which is the bottom left-hand corner. It shows how we treated diabetes a decade ago, 8 years ago, 3 years ago. And you can see people need more and more drugs to actually treat their diabetes. So, and that is indicating of a progressive disease. And it's also indicating that we don't think that the optimal treatment existed, because if there was an optimal treatment, you would need one drug and only one drug. In the upper right-hand panel, you see the US distribution of age, and as you can see, people are getting older, and with that, more will become diabetic. So it's just going to be a bigger and bigger problem. So this all is very important, because diabetes in itself is characterized by elevated glucoses, but the consequences are the 6th leading cause of death in the world, the leading cause of blindness in adults, the leading cause of end-stage renal disease, and a very important cause of amputations. So all these diabetic complications have very severe consequences for the individual patient. So I'm going to switch gear now to give you a glimpse of how the pharma industry looks at drug development for diabetes. Early on, we need about 10,000 compounds in what we call target to hit. So we make 10,000 new chemical entities. It takes 10 to 15 years to develop a diabetes drug if you have a target, which is the first problem. It takes about $1 billion. And we have different phases of development, which I'll get into: discovery phase, program phase, and then a product phase when you do your final development. In the early stages of development, we need a target, and this is where the genetics come in. This is where special populations come in, because the target is what we will target when we make a new drug, and that may be different in different populations, and it may be potentially targeted to the individual patient if we know what the problem is in the individual patient. The next stage of development is phase 1, and it is very common that when you do your first-in-man study, you actually go straight to patients with type 2 diabetes to see if the drug works. And so our proof of concept in man is actually as soon as we can do it, and probably within 1 year after we start testing, we would know whether we have a drug for diabetes or not. We use biomarkers very early on, so if we think that it works also on lipids or inflammatory pathways or inhibits hepatic glucose production or others, we use those in the first-in-man study. So we incorporate as much biomarker work and as much technology as we can in this study, simply because that study is $5 to $10 million. The next phase is maybe $50 million, and phase 3 is about $500 million. So if we can stop the development in phase 1, we save the $40 million or the $500 million